Good morning, everybody. Afternoon to some. Welcome today to today's Speaking of Oats session. My name is Eric DeBleek. I am currently working with Gray Millers and serving as the Vice Chair for the Oat Global Board. Oat Global is a public and private partnership committed to improving resilience, quality, and the value of oats by coordinating pre-competitive research, breeding, communications, and an extension on a global scale. Speaking of Oats is a collaboration put on by the Oat Newsletter and Oat Global, which has been organized by Charlene White with Agriculture and Agri-Food Canada and Dr. Jim Bradeen with University of Minnesota. So a big thank you goes out to those two. They've done a lot of great work here, putting together this series and helping coordinate all these sessions. Today, we will be using the chat function for any questions or technical difficulties. So please submit any questions that you have during the session via the Q&A function at the bottom. And after the talk, we'll hold a dedicated conversation or portion for the speakers to answer those questions. Our panelists today will be Kevin Smith, who is a barley and oat breeder at the University of Minnesota, and Walid Sadok, who is a professor with the University of Minnesota as well. They will be discussing improving heat stress tolerance in Minnesota adapted oats using eco-physiological approaches. I'll now turn it over to Kevin Smith to just start the discussion today. Go ahead, Kevin.
Great, thanks a lot, Eric. Well, I appreciate being invited to this series. Somebody who's been relatively new to oats. I've been taking part in it and have learned a lot. So hats off to the people who've organized this. It's been a really nice series. So as Eric said, I'm at the University of Minnesota. Most of that 22 years now that I've been there has been working in barley breeding and genetics. And for the last 6 years now, I have been working with the oat breeding program. We had an oat breeding program for a long time at the University of Minnesota. The prior breeder was Dion Stuthman, who was the breeder for over 40 years. When he retired, the program was idle for about 7 years. And then there's some interest in restarting the program, stimulated in part by an investment through PepsiCo to reestablish a public oat breeding program at the University of Minnesota. And so I got involved in doing that about 6 years ago. So it's been a really interesting challenge to get that program up and running. We have essentially a full breeding program now, from crossing all the way through to lines in advanced yield trials. We've started some really interesting, I think, research projects around traits that are important for the University of Minnesota program, particularly crown rust resistance. So we've done work with high throughput phenotyping to better measure crown rust resistance over time. And we're also doing some work on recurrent selection to develop non-race specific resistance to crown rust, which is another important objective we have. And then we've also looked at lodging, which is another big trait, and that work has been done in collaboration with some civil engineers at the University of Minnesota, using some engineering modeling which is typically used in buildings and bridges and things like that, but applying it to plants to think about how plants are structured and they respond to stresses like wind. So it's been really interesting establishing this program and getting started. And part of that excitement around the last 5 years has been working with Walid Sadok. So he joined about the same time that I started gearing up the oat program again. And he joined our department as a physiologist to bring that expertise to our department, which was desperately needed. And he's been a highly sought after commodity by many folks in my department to partner with, to help understand some of the underlying mechanisms for the traits that we're really interested in our department. And I was lucky enough to get to connect with him early on, so that before his plate was full. And so there's two major projects that I work with him on. One is on freezing tolerance in winter barley. So that's a project that we're working on to try to establish winter barley as a potential crop in the Midwest. And we refer to that as our cold barley project. And then the project he's going to talk to you about today is referred to as the hot oats project. So I like to keep things simple. So he'll talk about hot oats, and I'll just say one more thing. I think one of the things that I've learned from Walid over the past few years is an increased appreciation for the importance of understanding some of the underlying mechanisms for the traits that we're trying to understand, study, and ultimately improve through breeding. And breeders tend to be very practical sometimes, and you select for the trait directly, and that's how you go after it. Or if we're lucky, we have markers that we can use to make predictions, and we don't need to know anything about the genetics of the trait or the underlying mechanisms. We can do a lot of things in breeding without knowing any of that. But Walid, with the help of a few beers sometimes in the afternoon, has convinced me that there is a lot that can be understood and learned by knowing some of these mechanisms and then thinking about how that understanding can be incorporated into practical breeding to make progress on really complicated traits like yield or like stress-related traits like drought and heat. So I will let Walid talk to you about hot oats.
All right, thanks, Kevin, for the nice introduction. Thanks to the organizers for inviting me and us to talk about something that probably started as a beer-fueled discussion, but it ended up being a very interesting and exciting project for me. So, and for all of us in the group. So I'm a plant physiologist with a focus on crops, and I have really strong interest in understanding crop response to abiotic stresses. And this focus, the focus of this presentation is of course on heat tolerance in oats. So I'm speaking here on behalf of Kevin, of course, and also Jose Lopez, the postdoc who was still leading this work. Now, of course, this could not have been done without the tremendous involvement and commitment of many people in my group, in Kevin's group, in terms of measuring very complicated traits in the field. I'm going to detail those traits in the next slides, and also from Kevin's group in terms of managing those trials, taking phenology measurements, taking care of the seed quality measurements, and a lot of stuff. And of course, I would like to acknowledge the support of the Minnesota Department of Agriculture and also USDA. Okay, so I guess all of us here are familiar with the landscape of oat production in the US, but it's certainly worth reminding in a presentation like this. So the upper Midwest is a large contributor to oat yield production here in the US, and Minnesota specifically is a non-negligible contributor as well. And all of us know that there is an interest in raising production due to market demand, and Kevin hinted to the involvement of PepsiCo, and there is also other companies involved as well. And this is because of the uniqueness of oats as this cereal, which has quite substantive nutritional and health value, and also in terms of this crop being useful from a cropping system perspective, since it adds diversity to the kind of traditional binomial soy, corn rotation, and also sometimes including wheat. And there are advantages of that in terms of disrupting pest cycle. We know that that's an issue as well for other crops, and also maximizing, diversity and ecosystem services. We also know that there has been a sharp decline in oat production in the US, and there is quite the literature that kind of discusses the sources for that, and there are essentially three main lines of arguments. The first one of which is the shift in terms of how farm operations work. So basically farmers shifted from multi-purpose farms to highly specialized farms that focus on soybean and corn. And this is happening since the onset of the so-called Green Revolution. The pace of progress also is a major contributor, which is normal. We all know here that oat is and was a forage crop, and horses were large consumers, and mechanization replaced those horses by tractors and machinery, and also transportation-wise, that happened as well. And there's also something else that popped up in those investigations, is that farmers themselves kind of get a sense of that there is a decline in yield potential. And in many of those surveys, there's a mention of biotic stresses or biotic pressure, especially rusts. And I would like to stick to the environmental factor here as a contributor to this decline, but move on to the abiotic component of the equation. And what is worth noting here is that during that same window where the yield declined sharply for those economic and structural reasons, during that same window, we know that climate change has negatively impacted yields for many crops, and especially here, as we can see, for wheat and corn, which are crops cultivated in the upper Midwest as well. And I would like to take a quick step back here to kind of talk about what we think about those environmental or abiotic stressors that are driving this yield decline. So I'm going to start with the first one, which is really rarely mentioned, which is an increase in atmospheric dryness. And there are a couple of papers recently published that quite conclusively show that there has been an increase in the late 90s of what we call vapor pressure deficit, which is, as mentioned here, a measure of atmospheric dryness. I'm not going to dwell too much on it. I would refer you here to a quantitative systemic review that we conducted in the lab. And that clearly shows that even under wild conditions, in fact, in hydroponic studies, and most of these are like studies where water was not limiting. So increases in vapor pressure deficits are driving yield declines. And this is something to be concerned about. When we talk about climate change, we think, of course, about global warming, but it is noteworthy to keep in mind that the temperature is actually increasing at a much faster rate during the nighttime. In fact, 1.54 times faster during the nighttime. And these increases have negative and measurable impact on yields, and not only quantitatively, but also qualitatively in terms of seed composition, seed quality, etc. And of course, there is this increase in heat stress and high temperature stress event in terms of frequency and intensity. And our group with Jose and Kevin, we put together a review here that looks at the potential traits that could help mitigate heat stress with an angle focusing on transpiration. And you will see later in this presentation why this was our chosen angle. So speaking of which, so temperature, and specifically in the context of Minnesota and oat, there's a nice study published in 2011 based on, which is essentially an environmental association study where people look at statistical correlations between yields and environmental variables. And in this case, the study covered a window between 1996 and 2009. And the conclusion was quite straightforward, which is there are decreases in yield, statistically significant and statistically associated with warmer temperatures later in the season, which correspond to the reproductive window. And this is consistent with the broad strokes of what we know about the physiology of this crop, which grows optimally 2021C and reduces its growth at temperatures higher than 28C. There are a few publications that discuss this. I would like also to point out a very recent paper that reached the exact same conclusion, this time in Finland, of all places, covering, using a data set covering a much larger window, 76 to 2018. Just zooming in here on the abstract. And the conclusion is quite striking in that. So in this study, they looked at barley and oats, and they mentioned that temperatures above 28 degrees during the time frame of emphasis decreased yields of both crops, but oat yields decreased more strongly. And they called for introduction of new heat-tolerant varieties of both crops, but especially for oats. And this is Finland. So this is not South Australia. This is not, North Africa, where I'm from. So it's kind of confirms what those general concerns about adaptation of oats to warmer temperatures. And very nicely, Kevin introduced the way we think about these stresses and leveraging this eco-physiological perspective, which is relating the physiology to the environment. So this is the framework from which we look at these types of traits. We did not invent this. There's a wealth of literature looking at heat stress effect on crops. I pulled this up from a review paper by Prasad et al. And I want to just explain really quickly this figure here to illustrate one of these key principles. So here on the y-axis, you have the floret fertility as a percent of the control. For heat-stressed plants, 5 days here for wheat and sorghum. I think the temperature was 36 during the day and 26 during the nighttime for both species, and the control was something like 25, 15 a day and night, respectively. So the key thing here to note is that the effect of high temperature stress on, and the reason why they focus on floret fertility is that it's a very good proxy for yield. So whenever your floret fertility decreases, your yield for sure will decrease. So what we can see already from figures like this is that the effect of temperature on yield is not linear as a function of the phenology. So if you impose your heat stress after a certain window, which is anthesis, you will incur lower yield penalties, if at all. However, if you impose the heat stress during that reproductive window, then you will have like a kind of a valley of decrease in yield potential. So here it's kind of centered on anthesis, which is a very important developmental window. And the mechanism here, just again in the broad strokes, we think that high temperature stress reduces yield through a sink limitation. And here when we talk about sinks, and you already said that I'm going to talk about source, we talk about the fate of carbon. So the carbon and carbohydrate and other compounds that kind of fill the seeds and fill, make yield. So we think that the flower opening during anthesis exposes the external key organism processes such as anther dehiscence and pollen germination and stigma receptivity to heat stress just by virtue of opening the flower. There's obviously here things happening before anthesis itself during gametogenesis. And here the literature is quite convergent on the fact that high temperature stress has a variety and a cascade of negative impacts on a variety of processes that are driving male and female gametogenesis. There's a wealth of studies that looked into this as well. So to finish with this kind of framework, obviously we talked about the sink, though it's also important to talk about the source, again, in terms of carbon. And here there's a lot of literature as well that indicates that high temperature stress has an impact, a negative impact on photosynthesis, decreasing the ability of the plant to fix carbon dioxide. It's going to be those photostimulates to make grain. And just here an image that describes the type of damage that the chloroplast, the site where photosynthesis takes place, changes as a result of the heat stress. It's exactly the same duration. This time is for corn for similar heat stress treatment, and you see loss of integrity of the phytocoids, and those are energy transducing membranes. So photosynthesis will be capacity and efficiency will be decreased as a result of this. Okay, so based on this framework, our goal is to breed for enhanced productivity in Minnesota adapted oat under high temperature stress. And here we have a kind of triptych or a three-step approach that were nicely presented by Kevin again. So the idea is to start by first identifying mechanisms for heat stress tolerance. And here we have to strike the right balance between parsimonious hypothesis making and going deep down the rabbit hole, which is sometimes a geek, something that's for me very interesting. But again, this is translational research. So we have to, the craft here is to know where to stop in the research to come up with a trait that is meaningful for breeding purposes, which brings us to the second step here. So the translation of those mechanisms to target traits amenable to high throughput phenotyping, and then use that method to screen to mapping populations using physiologically informed approaches to identify tolerance genes and LEs and integrate them into elite germplasm. So for the sake of time here, I'm going to focus on these two steps here. So for this work, I'm going to quickly outline the methods and then the key results and the outcomes of some of the key experiments. We used for this genetic resource called the Minnesota Oat Founder Panel, which consists of 256 lines that were planted in different locations. And here, the technique we deployed is delayed planting. It's well known, well established. So we will plant those guys in the location at the normal planting date for that location, but then we delay the planting by 2 to 3 weeks, such that we increase the likelihood of exposure of the developmental, the reproductive phase to higher than usual temperatures. So that was one work. I will show you the key results from that work. The other one is a little bit more controlled. And in this case, we use what we call a core panel from this founder, identified by Austin Case, a former postdoc of Kevin's lab. And in this case, we focused on 30 lines that maximize geographic and genetic diversity. And these lines come from those different breeding programs here. I presume some of the attendees here are familiar with those varieties and those breeding programs. And obviously, these have to flower within a very tight window of time, because otherwise flowering will explain everything, and we don't want that. And for these genotypes, we deployed what we call the heat tent, which is I'm going to explain in a few slides. So essentially, it's imposing artificially in the field the heat, semi-controlled heat stress treatment. So in terms of measurements, I'm going to mirror here the kind of framework I illustrated in the introduction, which is trying to track traits related to carbon source, sink, and yield. So we measured gas exchange, namely transpiration, stomatic conductance, photosynthesis using photosynthesis systems, leaf anatomy as well. I don't have the time to dwell on that in this presentation. Also, we looked at canopy panicle temperature through different approaches using the photosynthesis system, also infrared thermometers or thermometer guns like this, and other tools that I will discuss in the second part of this presentation, which will have some sort of its own method section. And in terms of the carbon sink, we measured pollen viability and floret fertility. In terms of yield outcomes, we measured grain yield, its quality in terms of protein content, oil, and beta-glucans as well, because quality is paramount to very important for oats as well, as we all know. So heat tents, this is how they look like. These are our oat plots. This is Singer, who was an undergrad student conducting photosynthesis measurements here. These are other members in my group helping out with those measurements. These are the time courses of environmental conditions across the season. We'll illustrate these because they are very important in terms of context to interpret the data that I will show later. So this is for 2018 and then 2019. On the top panel, this is temperature, then precipitation, and vapor pressure deficit. It's important. I will not dwell too much on it, but I will probably hint to it a couple of times at this presentation. The black line here represents the mean values, and the gray area here it's illustrated daily ranges. Now, the most important part of this figure is the region, the period of time where we impose those heat tents. We did this between two key stages, between booting and heading, which is just the window before enthesis, where the damage is maximized, as illustrated in the earlier slide, and with this consistently between 2 years. Here I'm moving to the environmental conditions inside and outside the heat tents during those specific windows I just outlined. Again, the same years here: temperature, VPD, and PAR, photosynthetically active radiation. So as you can see, when looking at the temperature, we succeeded in obviously increasing temperatures as using those heat tents. And the good thing is that we did this much more strongly during the daytime than during the nighttime. In fact, the nighttime values were not significantly different. or very weakly. I don't remember exactly, but it's that we were very confident that our treatments really involved only differences in daytime temperature, which is important because we know that nighttime temperatures have their own effect, and we wanted to disentangle that. You can also see that 2019 is slightly hotter and actually drier when you look at VPD as well, which can explain some of the results, but not in a way that is inconsistent with the previous year. Final comment here on this slide is the similarity in light profiles inside and outside those heat tents. We have very similar patterns and profiles. We have a slight extinction of incoming radiation by roughly 22% in both years, especially for the very high light levels, or for the very sunny days. Okay, so in terms of the key results, I'm going to start with the delayed planting approach, for which we also leverage pre-existing data spanning two or I think 2 years before the project started. What you can here see very clearly is this negative, obviously, is highly statistically significant correlation between yields and mean temperature before anthesis, 18 to 19 days. Again, that key window where we think the damage on yield is maximized as a result of higher temperature. This is the average day plus night temperature, and this is the Tmax. So you see the same type of relationship here. So you can also even see things that confirm the literature here, in the sense that you start to see the decrease visually. Here we fit the linear regression, but maybe there's a nonlinear thing happening here. But for sure, starting for temperatures higher than 20 degrees, you start seeing the decline in yield. Now, this is therefore a confirmation that the problem is important in Minnesota, that we strategically did the right thing in terms of focusing on that window just described. And the other thing, don't have the time to show this, but these correlations are much weaker pre-anthesis. So at least in Minnesota, we think that the susceptible window that really causes yield declines is flower development rather than seed fill per se. So moving on to the outcomes of the heat tent experiment. So on this slide here, I show the results in terms of impact on yield and its quality. So here the gray part represent the data of 2018, the open part represent the data from 2019. Every time you see a blue box and whisker, that represents the control plots, and the orange plots represent the heat stressed plots. And clearly here you see that there was a consistent decline in yield as a result of the heat stress treatment, and this is good for us. So it means that this experiment has succeeded, and the declines were quite similar. We have each year an increase in protein content as a result of the heat stress, and this is actually consistent with what literature says in some species. Same for oil content, it declined. Actually, the opposite. I said the same in terms of what the literature says, but we have also a decrease in oil content as a result of heat stress. So clearly, this shows that heat stress will also influence quality traits in oat, in addition to grain yield. In terms of beta-glucans, we have a kind of similar trend between year 1 and year 2. It was significant in year 1, but not significant in year 2. So, as I mentioned, we are really interested in this kind of source sink framework, and we wanted to get a sense of what drove this yield decline. So we measured floret fertility, as I mentioned, in material methods, and here clearly, as you can see, is that the heat stress treatment decreased quite substantially floret fertility in both years in a consistent manner. And rather surprisingly, the effect on pollen was not consistent, kind of decreasing the first year, like the fertility decreased kind of, I mean, statistically significantly for sure, but there was a lot more variability in year 1. In year 2, the trend was non-significant, and if anything, it was the other way around. So floret fertility was explained much better than pollen fertility, the yield declines. And the consequence of that is that floret numbers decreased and grain number also decreased. decreased quite consistently with the yield outcome. So in terms of the source side of the equation, and here I'm going to show the meaning photosynthetic rate, which is on the y-axis here. It's the same type of figure. Again, there is a decrease in photosynthesis as a result of heat stress in a consistent way in both years as well. And this indicates some evidence for sink limitation, not necessarily very strong, but it is there. It's strong enough to be captured statistically. So there is a decrease in input of carbohydrate for grain, for seed fill as well. So something really important, and this is what connects to the paper I mentioned, dealing with heat stress and transpiration and temperature, canopy temperature. So this is a result that was quite important for us in this project. So we measured gas exchange, meaning not only photosynthesis, but also transpiration, represented here on the y-axis. And what you can see here in both years, under the heat stress treatment, the transpiration increased. And we thought that if this was the case, it should also be captured in our canopy temperature data measured using the gun thermometer. And indeed, it was consistent with that. So this is leaf temperature depression. So the higher the value here means the cooler the canopy. And indeed, we have an increase in canopy cooling as a result of the heat stress. So two messages out of a figure like this. First of all, that we very likely did not have an interaction with soil moisture deficits. So we're only looking at heat stress, because if we would have had soil moisture deficit, then the transpiration would decrease here, because it would be drought, and that's one of the hallmarks of drought. And the other thing is that those genotypes are trying to cool themselves as a result of this heat stress event. So we then started to look at the significance of this in terms of impact on yield. So we undertook a simple correlation analysis between all the traits. We did this for the control and for the heat stress configuration. For the control, there is very weak correlations. Some of them are expected between grain number and yield, stuff like that. So I'm going to focus only here on the heat stress correlations. And what you can see, I would like to draw your attention to the yield column here, and you see the correlation, the negative correlation with protein as shown, positive correlation with floret fertility, floret number, grain number. That's expected, but also very strongly and positive correlation between yield and canopy cooling traits such as stomata conductance, transpiration, and leaf temperature depression, meaning that higher yields under heat stress were associated with higher stomata conductance, higher transpiration, and therefore higher latent cooling. And we confirmed this measurement in this idea in 2019, this time by measuring panicle temperature, which we did not do in 2018. So basically that means that what the cooling through the leaf is also impacting the panicle itself. We also think that panicles themselves might be transpiring. It's still an idea out there, which could also help explain this as well. We know that they are photosynthesizing, so why not transpiring as well? So now we have an idea, like I mentioned. I was tempted to go a little bit deeper into the kind of mechanistic aspects of this, but we have a trait that is actionable, which is canopy temperature. So this is where we decided to kind of try to move with this to develop a high throughput phenotyping system. And for this approach, we selected 8 oat genotypes that contrasted highly in their yield response and floret fertility here during the 2018 and 19 heat tent experiments. So these were carefully chosen to reflect the breadth of those responses. So we had champions and losers in that regard in our selection. And then we decided, so using gas exchange systems or gun thermometers is a time-consuming approach. So we decided to deploy what I call thermal high throughput phenotyping approach using a thermal camera that is mounted on a drone. And we did that, but we also did in addition to that, a couple of kind of other approaches in parallel. So on flight days, we would use this approach of thermal cameras mounted on drones. On non-flight days, which are days where the wind speed is too high, higher than 15 miles an hour, where we cannot fly this drone, and these days are typically sunny, etc. We use a handheld thermal camera. And for both of these, we have what we call ground truthing, which is basically measuring temperature as experienced by the plants themselves through thermocouples connected to multiplexers and double loggers. This is very important because what we see here are temperatures, after, these are remote sensing basically techniques, and this is a proximal sensing technique, and that needs to validate the former. So this was developed in a large way by Jose. This is Jose, he's driving the drone here. This is Bishal, the former postdoc, who was helping out as well. This is an image, giving you the sense of the altitude for those measurements. And this is like in a nutshell how the field looks like from the thermal perspective. And there's a lot of data analysis here that has been developed by Jose. He developed R scripts to do those types of things and extract data for each plot, and from those data he could compute temperatures. And we're still working on this, and we'll be happy to discuss some of those aspects in the discussion. But the key idea here I would like to kind of present is the consistency between these different approaches we used, which is very important prior to looking at the data of the experiment. So first of all, the handheld thermometry was really quite consistent with the thermocouple data, as you could see here in this figure. And these were made over different dates in the field last summer. This figure represents the relationship or the correlation we found between the handheld thermal camera and the data resulting from using the drone mounted thermal camera, which has slightly different characteristics. So there's what we think is a reasonable agreement which is between the data sets. And the reason why we're not hitting the one-to-one regression here is that we think, so probably other reasons. So the handheld images typically took place 30 minutes after the flights were completed for obvious logistical reasons. And because of the manual aspect of taking those images, it took 30 minutes to collect images versus roughly 5 minutes via the drone. And this is important to know. So that means that for some images, you have a 1 hour delay between what you measured over the drone and what you measured at close range. And between these two times, key conditions would have changed because transpiration is a very dynamic process. So wind speed could have changed, solar radiation, cloud cover, all of these things add to the error. But we were reasonably happy with the correlation to decide to use the drone. And we flew the drone on the same critical window I mentioned, which is between booting and heading on those 8 genotypes, and examined the temperature as captured, canopy temperature captured through that process with the floret fertility values based on the 2-year heat tent experiment. And this was the outcome of that experiment. So on the y-axis here, you have the floret fertility during the heat tent experiment as a percentage of the control. And this is the drone canopy temperature in that key window I mentioned in 2020. So obviously the key thing here is that we have a significant and reasonably robust correlation between the two, and in a way that is consistent with our hypothesis, meaning that the champions, the genotypes that minimize floret fertility loss in response to the heat tent experiment are actually the ones with the cooler canopies during that window in a different year. So while the opposite is the case for the kind of the losers, the ones that are very sensitive to heat stress. So this is for us is encouraging, is consistent with our framework, is consistent with literature, is consistent with the biophysics of transpiration and canopy temperature dynamics. I'm gonna stop here because this is what we're gonna leverage for high throughput phenotyping. But before leaving, I would just like give you 4 takeaways from this presentation. So I hope I was able to show that heat stress is a major oat yield limiting factor in Minnesota and other temperate regions, presumably. Doesn't necessarily require excessive temperatures to reduce yields due to the high sensitivity of the crop. Those yield losses, at least in our case, are largely due to florid fertility losses, and canopy, if not panicle cooling, appears to be a promising trait to minimize those losses. Finally, we have identified a clear pathway for a high throughput protocol to identify genetic markers associated with cooler canopies and better heat stress tolerance in oat. With that said, thanks again for the invitation, and please, I would be happy to have a discussion with you. So please feel free to ask any question. Thank you.
Thank you so much, Walid, and also Kevin. That was a really interesting presentation. And I know actually that Jim has a question. So did you want to start us off there, Jim?
Sure, yeah, excellent presentation, really interesting. In your heat tent experiments, I was really intrigued by the observation that heat stress led to greater protein content. Do you know anything about the protein profile? What proteins are actually increasing as a result of heat?
Yeah, that's a great question. The short answer is that I don't know, and this was not something we thought about in great detail before the experiment. But I would expect some changes that relate to, like for instance, when you increase temperature that way, you could also have an impact on processes that can denaturate certain enzymes, can impact certain biochemical processes, and these can have the outcome of which would be different, like a protein accumulation. This is the accumulation of the protein. This is something that has been observed also in response to drought stress. I was trying to recruit some mechanisms that I know exist for drought stress, but they're not necessarily the same. So that's why I'm stopping. But this is definitely something we would like to, I personally would like to go after if certain protein profiles are important for the industry, for instance.
I mean, the general phenomenon is in part at least due to the ratio of carbohydrate to protein, right? So your protein concentration goes up because probably your carbohydrate is going down in that heat stress. So I think at least that's part of what's there, but I don't really know anything about the specific protein, like which proteins are being affected.
Sure, yeah, that's true.
Great. Next, Bruce Roskins has a question. Were temperature and variety the only 2 variables, or was there any other potential limiting factors such as disease, etc.?
I don't remember in the notes or in the discussions anything about disease. I don't think we have any interaction with abiotic stressors in this experiment. And it was kind of really a concern of us to really make sure that an experiment like this, we did actually a lit review before starting really the project, after having the funding, to really ensure that we have only the temperature, temperature during the nighttime being the major kind of structuring treatment, but we did not come across any pest or pest-related issues in this experiment, and thankfully so.
Okay, and Christian Azar would like to know, were the losers shorter or were they tillering less than the winners?
I don't know about that. I think the simplest explanation is that they probably at least from the perspective of our kind of simple framework, they probably they're not efficient in cooling themselves compared to the champions, or that's for sure something that shows up. but there might be other processes as well and that could be involved. But I don't know about tillering or height.
Yeah, and of course, one of the things I always think of is the waxiness as well of different varieties and how that relates. Bruce Ross taught me about that.
So that's a very good point. We did not look at that. That's a short answer. But in that review paper we put together as part of this project, we really discuss the involvement of waxes because they have their melting points, and if the temperature reaches a certain level, you start having evaporation, and that evaporation is helpful to cool the canopy. And we speculate about this being actually a mechanism. So the composition of the wax or the genotypic variability thereof could be one of the players there. So the consequence of what your comment is that when we talk about transpiration, we see that the canopy cooling co-trans with stomata conductance, but we cannot exclude changes in wax's properties, the treatment. But it's a very good point.
Thanks. Okay, so Axel Garcia says, nice presentation, Walid. Increased air temperature increases VPD. How much reduction on stomatal conductance are we talking about as for today? And what about in the future?
Yeah, good point, Axel. Actually, we've seen an increase in stomatal conductance in those experiments. So that means that probably the VPD range was not, the values were not as high enough, and the soil moisture was not limiting to result in a decrease in stomatic conductance. So what was the other part of the question again?
So what are we talking about, today compared to what will be happening in the future?
Oh yeah, well, that's so that's the other leg. So climate scientists project that VPD will continue to increase in the future, and those models try to kind of project the consequences in terms of stomatic conductance. So parsimonious assumptions indicate that stomatic conductance might decrease as VPD increases, but I would assume that all of that depends on the soil moisture, the local hydrology, and also on the community assembly, the diversity in genotypes. Genotypes of the same species do not behave the same way. Different species do not behave the same way. So it's still up there. There's a huge error bar on what's going to happen in the future in terms of stomatic conductance. That's what's, by the way, one of the drivers of that review paper we put together about VPD. So, yeah.
Okay, thanks. And Catherine Howarth would like to know, did the grain size decrease with heat stress?
I should know about this. Kevin, do you remember? I don't have, it's somewhere in the data if we measured it, but I'm not even sure that we measured a grain size.
We do have measurements, but I know that Jose presented that, but it's been a long time since I've seen that.
Yeah.
Something to follow up on.
Yes, because it would provide a nice kind of validation or testing of the effect on reproductive development, right? So it's an important point for sure.
So Jean-Luc Yannick says, oat is sensitive. The mechanism you propose mitigates the condition, too hot a canopy, but not the sensitivity. Would there be mechanisms to mitigate the sensitivity?
Yeah, that's a great point. There probably are, so when you look at the literature, they're like, so the damages, we can speculate here. I'm speculating without the beer, so be careful. So we know that heat stress damage, damages the pollen grain itself, not necessarily in our experiment, but in other crops, it's the case. The stigma receptivity, anther opening. So I would speculate that molecular or physiological mechanisms that enhance the resistance of these steps towards high temperatures would influence that sensitivity. So lipid composition comes across really frequently in those papers. So I would speculate that playing with genes that regulate the lipidome, for instance, would be one of those approaches. But presumably, I would assume that like biochemistry and molecular biology and some gene editing would be useful to kind of entangle those things. But yeah, there are a lot of things to entangle in that realm for sure.
Great, thanks. And from Atakir Raman, if I understood correctly, plants were under heat tent kind of shelter. Then how did you manage water irrigation requirements? Did it also affect your results?
Yeah, that's a fair point. As I mentioned, I don't know if I mentioned that I think we irrigated once and prior to putting the heat tents. But for sure, that was one of our concerns. But we did not impose an irrigation system during the experiments for a couple of reasons. But the short answer is that the data does not seem to suggest that we had a strong interference, if any, with the soil moisture deficits because of the way some other conductants behaved. But there is, we cannot very firmly say that there was absolutely no time in the day where plants experienced a mild water deficit or a transient water deficit.
Mm-hmm. The next question is from Hamid, and I'm sorry, I'm going to butcher your last name, Shuldel Maganlu. Oh, I apologize. I don't know how to say that. Did you have any way to account for the confounding effect of the accelerated development caused by delayed sowing in your analysis?
So say it again, the question about, so the accelerated development, right?
Yes, caused by delayed sowing.
I need to look at the data about that. I'm not sure there was that much. So, I mean, even if it existed, right? It's, I guess I'm wondering what would be the problem in terms of interpreting the data. But I need to look into that idea and parse it out. It sounds interesting. So, but I'm not gonna venture and give you like messy answer to that. So. Okay,
fair enough. And Elizabeth Zechner would like to know, did you see differences in husk thickness or hull thickness? Thicker husks by higher temperatures combined with smaller grains maybe. So this relates a little bit to Catherine's question, I think, too.
No, but that's an interesting idea. Yeah.
Okay.
Yeah, we did not measure that, so we wouldn't. Yeah.
We have plenty of ideas based on the results for sure, but those also are very useful. So thanks for that.
Great. And Juju says, great presentation. I wonder if there's any research plan for dissecting the genetic mechanism of regulating protein or oil content under heat stress.
So Kevin, what do you think?
Yeah, well, I mean, as a first step, we could use the founder panel, has a set of markers, GBS markers associated with it. So we could start with a sort of a GWAS type approach based on the multi-environmental data. But we only have quality data for several of those environments. So it would require possibly getting some more protein data from those environments, but that might be a first step.
Could an RNA-seq approach inform this?
I would think so, yes. I haven't really thought much about how to set up that sort of an experiment, yeah.
feels like it might be easier to start there. But
Yeah, no, I mean, as a forward experiment, I'm just trying to think about, is there something, do we have historical data that might help us to think about that? But yeah, that would make sense.
Right. Okay, and there's another question from Christian Azar. Did you measure canopy cover in each genotype? We would expect that bare soil would increase temperature around the plants if the canopy is not covering the ground well. Are upright plants less effective in fighting heat since they let the sun reach the ground? Were the losers covering the ground less than the winners?
I had to say, I don't know, just but like trying to give you something to chew on here. Anecdote, say again?
Sorry, it kind of relates to those South Dakota type lines too, with a lot of wax. They tend to be very upright and thin looking.
Yeah, I anecdotally, I noticed, the reason why I say anecdotally is that I don't establish a relationship, but I took a lot of pictures when I used to walk in the plots, because there was clearly a variability in leaf width and canopy kind of architectural types of traits. Now, the intuition, when you see that, you start thinking about energy balance inside the canopy and energy transfer as influenced by canopy structure. So we did not go into that realm, but we are interested in that because incidentally we're interested in sheath physiology. We found that the sheaths actually do photosynthesize, do lose water, so that can actually have implications in terms of the canopy microclimate. So I would assume that canopy architecture would more or less allow more light to get into the sheaths, and therefore there's some feedback loop into what the sheaths can do in terms of transpirations, stuff like that. I think it's a good idea to look into that component, into that big box of the canopy structure and the energy balance inside the canopy and coverage as well.
Do you know, Walid, do you know if Jose was able to estimate canopy coverage from his aerial drone images? Because that might be one way. I'm not sure if it was the right phenological stage to get differences in that.
Yes, I think he flew the drone a few times to be in order just to track canopy growth. For sure, we did that. He did it for certain purposes in terms of like testing the drone but we might be able to look at that data and see if there's some co-trends of the canopy kind of growth rate kind of has a role to play in this. But yeah, it's an interesting idea.
Mm-hmm. Yeah, that's really interesting, and the sheath thing too. So Jennifer Mitchell-Fetch now has a question. In the future, would there be an opportunity to evaluate a connection between upper plant structures and root depth or size with respect to heat tolerance?
Short answer, I think yes. Because, you know, there are trade-offs to this, and this is the main angle of that paper, the third paper. So here in Minnesota, we are lucky enough to be in an environment where I don't think people in the back, you know, the head of the people drought is like this threat, like soil moisture deficit, at least the soil moisture deficit part of the equation, not the atmospheric one. So plants can't afford basically this cooling strategy without marginal costs in terms of the water budget available in the soil. However, in other environments, probably southerly, like more to the south, or in environments where the soil is not very deep or very highly draining, root depth is going to go, you know, especially if the roots track the water table. So basically, root themes will be related to heat tolerance of the canopy. So if you have a root system that is able to track the water table and make the plant afford losing water, that if you don't have that, then especially in an environment where you have soil moisture deficit, then the canopy cooling strategy would be risky from a water budget perspective. And we speculate that plants will have to make a decision. Functional ecologists, people will tell you that probably plants will sacrifice water because the risk of carbon starvation is more important for them. So they don't want, so that's what from the ecology realm, but for crops, there's other pressures, other processes taking place. So definitely root stuff is important here as well in terms of water acquisition.
Interesting. There are no more questions in the Q&A box, but Weekai did have actually some comments before. Peter Scott from New Brunswick has indicated to me that the oat yield has been decreasing in their provincial trials while new cultivars tend to yield higher. So that's more of a comment. And this is Week-Ian also said, I looked at mean yield at the Quebec provincial trials from 2010 to 2020 and did not see any trend. So it's comments from Week-I there. And there is now another question from Sepper Naragi. Great presentation. Did you evaluate thins percentage?
I'm sorry, say it again.
Did you evaluate thins percentage? So the percent of really thin oats in the samples.
We have that data, and I can't remember what, Jose was doing the analysis. We have that data, but I don't know if there was an association.
Great. So lots of great work and lots of great questions and things for you guys to do too. So.
No, these are very useful. Actually, they're kind of making me, churn ideas around and kind of, figure out the best next step. Obviously, the high throughput phenotyping and the genetic analysis would be a good way to go. That's for sure. And the quality aspect for me is super exciting as well, because the industry is highly interested in that. The change of the grain profile is paramount here as well.
Of course. Actually, we do have another question now. We're a little bit over time, but I guess I can go ahead with this one. From Jim Dick, would a semi-leafless oat benefit cooling?
I'll be honest here, I've never heard about I'm discovering oats. Okay. Yeah. So was that a leafy or less leafy oat?
To me, that suggests less leafy. Jim, did you want to jump in? Jim Dick, if you wanted to add something to your question. He's an oat breeder with Oat Advantage out in Saskatchewan here in Canada, where I am. Oh, less leafy. There's our clarification.
Okay, I can speculate. So I would say, like a very, very simple argument, if the leaves are the source of cooling, I would presume that like is an optimal number of leaves for cooling or also for radiation interception and so forth is important. I would actually, if there is an idea I would like to inject here as well, like the canopy, I think we think that canopy, the panicle itself might be cooling itself. It's happening in rice, and there is like very impressive data that shows like cooling of like upwards of 5-6 degrees for like some genotypes can do that. So we used leaves as proxy, but maybe the panicle themselves do that.
Jim's added a comment here. I have seen oat lines with good panicle structure, but a much reduced leaf. So, yeah. Oh, and then what about then more upright leaves? So this, yeah, we touched on a little bit before. So still the quantity, but more air movement. So yeah, more air movement could be a factor there.
Yeah, upright leaves. That's so there was work like a couple of decades ago on that. So the theory is that upright leaves help increase canopy level photosynthesis, and maybe as a result, higher rates of gas exchange and presumably from the sheath. So maybe the leaf angle lever would play with canopy transpiration in a way that could increase that cooling effect. But again, it has to be a function of the water availability regime in a given location, right? But yeah, it's an interesting idea.
Okay. Jim says thanks for the discussion. I do see in the chat, there is another question here, actually about roots from, oh dear, Sajivan Radha Sivarajan. I'm sorry, I didn't say that properly, I'm certain. Great presentation. Is there any way to understand or correlate the below ground, i.e. root influences and heat stress? So those genotypes have a better root system and lesser canopy have higher tolerance and vice versa.
Yeah. So as I mentioned, the simplest explanation about like that can connect, like the simplest link between roots and canopy cooling is through the water acquisition angle. And not necessarily just depth, maybe like the ability to kind of mine for water, you know, it could be something else than just deeper rooting. The other link I would think of is if certain soils, so when we don't think about this too much, but when you have high temperature stress or heat stress, think a lot about air being warmer, but what the soil can get warmer to such an extent, at least in certain layers, upper layers, and maybe that would increase root respiration, and that's carbon that is lost that could go in the seed. So that's a potential link there. So genotypes with different root respiration sensitivity to high temperature could result in better yield because of this less of the allocation. I know that's a problem in horticultural crops because they grow them in pots, and the temperature in the pots can get like to 35-40 C. So that's a problem for them, and they're looking to that. They work on trying to understand soil thermal conductance and those types of processes that affect roots. So these are basically the two kind of connections I would see.
Right. You have a lot of work to do.
It's a never-ending thing, but again, that's the trap and the thing that like we want to find traits that are can help breeders and people doing management to move the needle. So it's kind of finding a needle in the haystack, so to speak, as well. So
right, well, that's excellent. I don't see any more questions. Actually, I wanted to give a quick shout out to Keith Armstrong here because he's in New Zealand where it's after 4 o'clock in the morning, I believe. So thanks, Keith, for joining us live today. And I guess with that, maybe Eric, I can pass it over to you. And should I show the final slide?
Yeah, that would be awesome. Thanks, Charlene. Just a quick wrap up today. I want to thank our panelists. I want to thank everybody who submitted questions and just everyone who attended here today and participated in our session. Charlene has put up a slide talking about the next Speaking of Oats session. There will be a date and title to be announced here in the near future, and we'll have one hopefully coming up in June. So, that date and information will be put out in the next couple of weeks. So look forward to that, and we look forward to having everyone at the next Speaking of Oats session. To those in Canada, I hope seeding finishes up well, and we get some rain and hopefully not too much snow out in the prairies this weekend. So for everybody else, I hope you have a wonderful weekend and the last little bit of your week, and take care. Thanks for participating today.
Thank you.